So in this chapter, like I said, we're going to give the background for the rest of the semester. We're going to review what we're going to be doing, taking a look at the biomolecules, reactions, and end on energy and organization. So we're going to then review much of this material again later on in the semester. But before we begin, why are we here, right? Why are we in this room? Of course, to study biochemistry. But really, we want to understand the chemistry of life. What goes on in our cells, and why does it go on? So we first have to have the knowledge before we can go into the pathways. So we're going to study biomolecules and their properties, and then go into the processes or pathways. This is a fraction of the material that you would cover in a third or fourth year class. So if you guys are going on in your studies, you may have to take another biochem class, but you'll find it a lot easier. So this is a nice way to begin. So this chapter is broken up into basically what I see 4 parts. What we're going to do is first review cells very quickly. The second part is we're going to review organic chemistry. So we're going to take and look at different PowerPoint slides for that. We're then going to have an introduction to our biomolecules and the reactions. So we're going to learn the organic chemistry and then apply that to our biomolecules. And then we're going to study energy and order, because the energy behind the reactions is going to be this background noise that we're going to be paying attention to as the semester goes on. So really, what is life? What makes my cup not alive and me alive? So again, the inanimate becomes animate. So we are going to be moving around, breathing, growing, and you can define that. But there's some things that are different about things that have life. They're very complex, right? You'll see the complexity in the 3-dimensional molecules in the reactions. The amount of organization is amazing. If you take a look at the structure of the cell, I think it's quite wonderful that it has evolved to be that way. And of course, it's based on cells, and it's information-based. So how do computers work? Anybody ever take any programming? No? Ones and zeros, that's right. But we don't operate on ones and zeros, right? So we will operate on the information held in 3-dimensional structures. How a substrate or a reactant fits into an enzyme, or how 2 biomolecules fit together. And so I don't worry about computers taking over the world until you start to see 3-dimensional molecules start coming together. Then you better run for cover, right? Because then they'll be able to be very complex. But with ones and zeros, there's a limit. They might beat us at Jeopardy, right? But they're not going to be able to really solve the complex problems and evolve the way cells can evolve. And it evolves. And we've seen evolution in our own lives. What is some evolution that we're seeing in a matter of years? Yes, Elijah?
Viruses,
bacteria, right? Where MRSA, right, has evolved. And so we can see evolution not in thousands of years, but in tens of years. Okay? So we're going to talk about that. So this is minimally, and we know that it's more than this, but this is a good start. So really, life occurs in cells. I'm going to go quickly through this material because I'm not a biologist. What I want to do by teaching this part is sort of place us where are the reactions happening, and that's why we're covering this part. Where are the reactions? The reactions are going to be happening basically in the mitochondria and the rough ER. And so we're going to talk about those afterwards, but we have to really know a little bit about cells. So most of you guys have taken a lot of biology, right? Yeah, so this is, back in middle school maybe you learned this, right? Prokaryotics and eukaryotes, right? So we know prokaryotes don't have a nucleus. Eukaryotes do, but it's a little bit more than that, right? It's a little bit more than just not having the nucleus. The eukaryotes can be multicellular. They are a little bit more complex than the prokaryotes. So we'll talk about the differences between them. But let's talk about prokaryotes before. You can either have bacteria or archaea. Bacteria are very diverse, all right? E. coli is probably the best studied bacteria. In my biotech history, back in the 1990s, when biotech really started out, they used to use E. coli to grow up the biotech drugs. And it was really hard to work with E. coli. They were great because they grew up really quickly. But what they would do, the gene that they would put into the E. coli to make the biotech drug would have the protein in the cell and something called an inclusion body, like a big glob of that protein. So when we would try to get the drug out of the cell, we'd have to break open the cell and spit out all the guts and then later purify the protein from those guts. And that was really hard. In modern times, we use a Chinese hamster ovary cell, or a CHO cell. Anybody ever heard that? That's for if you're ever taking a biologic drug, most likely has been grown up in a Chinese hamster ovary cell, which is obviously a eukaryote. If you look at it funny, it will burst open, but if you treat it well, you can have the biotech drug be spit out from the cell, and it's a lot easier to purify. They don't grow as well, but it's a lot easier to get the drugs out. So, yeah, I used to work with E. coli. And our archaea are increasingly a area of research because they can work in very extreme conditions of temperature, salt, pressure. They're called extremozymes. The bioremediation of oil spills, they can use archaea for that. Toxic waste sites. All right, so let's take a look at the structure. It's pretty small. They can move with the flagella, and it can be gram positive or negative depending on the cell wall. And they have cell walls, plasma membrane underneath, and this circular DNA. All right, the circular DNA allows it to become to evolve very quickly. And so the biggest difference is that we don't have organelles with prokaryotes. So what made that E. coli really hard to break open was this cell wall. And it's above this plasma membrane. And we're going to talk about later on when we study membranes why this cell wall is so tough to open. We would have to put these cells through like an incredible amount of shear in order to pop them open. And what happens is you have a cross-linking of proteins and sugars on top of the cell, and it really makes it a tough cell. So this is a picture of what makes it so tough. So you can see this is the inner part of the cell, and this is the inner membrane, and then this is the cell wall. So directly underneath that is the plasma membrane, which we are going to spend a great deal of time on in this semester later on. but you can see on the outside we have some sugars to detect what's in the environment, and it has channels to let things in and out because the lipid bilayer doesn't really let things in and out. So we'll go over later on the semester in detail a plasma membrane. Aren't you looking forward to that? Can you not wait for that? Inside that membrane, we have cytoplasm, which is just goo, right? It's goo made of, we can have our circular DNA, chromosomes, plasmids, small circular DNA, and then ribosomes. Ribosomes actually give our prokaryotes a grainy appearance. If you look at them under a microscope, the little grains are actually ribosomes. What happens at ribosomes? Does anybody know? Protein synthesis. So we are going to study that in depth. And then you can have appendages. So again, what's the difference between pili and flagella? So pili, am I saying that right? Am I? I don't know. Yeah? Good enough? Yeah, all right. So these are more fine hair-like structures, all right, that allow attachment to food and maybe a host, where flagella are flexible protein filaments used for locomotion. All right, let's move on to eukaryotes. So how do they differ? You have the presence of the nucleus, much bigger, more complex, and you can get be multicellular, so they can come together. And as you can see, it is really complex. So here we have a cross section. I'm sure that you've seen. So really, we're going to spend a lot of time in the mitochondrion and these little dots here. These are the ribosomes. All right, so what we're going to do is go through each part. But you can see maybe a question that I might ask would be, discuss the differences and similarities between a prokaryote and eukaryote. You guys at this point should be able to sort of predict what the questions will be on exams. So you can see it's very highly complex. And these organelles actually interact. All right, so let's go through each. Again, the plasma membrane, you got to have that for life. Okay, you've got to have a membrane in order for a cell to exist. Actually, what do you have to have for life? What do they look for in other planets to see if life is going to happen? What did they find on Mars and everybody got jazzed?
Water.
Water, right? You have to have water in order to have life as we know it, right? So, but the next thing would be a plasma membrane to have a cellular life. So again, it's going to keep everything inside, and it's going to be composed of this bilayer. It's phospholipids. So this phospholipid has a lipid part and a phosphate part. The phosphate part is charged, and the lipid part is uncharged, and or hydrophobic. And those hydrophobic parts come together, and the polar parts stay on the aqueous parts, the parts near water. Again, it's going to provide the shape, strength, although it's not very strong. It's easy to move around, and it's going to protect the cell. And we stud the membrane with lots of different things. We're going to put proteins that can be channels. We can put receptors. They can be the sugars. Whenever you see sugar on the surface of something, think, ooh, it's looking for something. It's going to interact with something. We're trying to look for a signal. And then you can have the glycolates, is that carbohydrate layer. Again, the nucleus contains DNA, directs its synthesis, and it's actually produced from a double membrane where you have pores where things can get in and out. And the chromatin fibers are DNA wrapped around histones, which are, it's kind of neat how this packs up. Because DNA is really negative and histones are positive. So you can't, DNA would repel itself quite a bit. So in order to stop all that repulsion, it's evolved to wrap itself around something that's positive so that it can be compact. Pretty smart. And so we have RNA synthesized from this DNA. All right, we have two forms of the endoplasmic reticulum, the rough and the smooth. The rough is where we are going to produce proteins, and the smooth is lipid synthesis. Again, what I want from you is just the main function of each part, which most of you probably already know, but just review that. The Golgi is our distribution center. It takes in new materials on the cis or inward face, processes them, and then releases it to the cytoplasm on the trans or outward face. We're not going to be here, all right? I'm not really going to cover anything that goes on here. Lysosomes are responsible for digestion. They have basically it's the like a trash processor. So it's going to break things down because it has digestive enzymes. So it's going to be breakdown of any material, worn out or unneeded components, and so it's going to take care of things. It's cleaning up. Macrophage lysosomes eat up damaged cells or foreign organisms, so it's really important that you have them. Peroxisomes, I don't have this on here, are organelles in which contain oxidative enzymes, which can detox our liver and kidney cells of ethanol. In plants, they are responsible for photorespiration and metabolism. So that's usually where you see them. Mitochondria. This is where we're going to be. We're going to spend a lot of time here. It's where we produce energy. So really, I mean, this semester we're going to take a molecule of glucose and take it down to ATP and carbon dioxide, and it happens in the mitochondria. So we're going to spend a lot of time in here. It's where aerobic respiration. What does that word aerobic mean to you? With what? With oxygen. So that word means with oxygen. In ATP, you're going to hear me call it our money molecule. All right, that's our energy molecule. We'll talk about possibly why would the matrix contain DNA, right? But it's double-walled and it has intermembrane space in between the two walls and then the matrix inside the middle one. And then we have the electron transport chain and ATP synthase that lie along the inner membrane. And this is what it looks like, a little kidney bean. And those of you that are athletic, maybe run, do any other cardiovascular, will have more mitochondria than cytoskeleton is the fibrous stuff of the cell. We have three different components: microtubules, microfilaments, and intermediate fibers. And you have maintenance of shape, movement of organelles. The difference between these three are their size. Microtubules are found in structures that need support. So axons and mitotic spindles, right? So they're about 25 nanometers in diameter. Microfilaments are composed of actin, and they are involved in ameboid movement. And there's various compositions of intermediate fibers. When I was in graduate school, I was very, very familiar with microtubules because I did anti-cancer research in graduate school a long time ago. Old-style cancer drugs, Taxol, which is still being used today, vincristine and vinblastine, they work to stop cell division. And the way they work is they stop or either permanently keep tubulin, the protein tubulin, from polymerizing into microtubules. So what I would do is there's like 10% of the soluble protein in brain is tubulin because of the axons. And so what I would do is I would get a thing of donuts and drive to my local kosher slaughterhouse and get 12 cow brains, fresh, the open brains, and I would wipe them up, bring them back to my lab, and isolate the tubulin from that. And we figured out by my group in graduate school doing that, we saved our lab $60,000. We would have had to order it. So it took us about 3 days to isolate enough tubulin to do our research, but we saved $60,000. So it was like $500 worth of chemicals. But what we would do is we would polymerize them and depolymerize them and sort of centrifuge things out, and that would purify them out. But then I would take a look at different drugs. Half of my group would synthesize drugs, and my half looked at the interaction of those drugs with the tubulin. And so I would do various experiments looking at the kinetics of the binding, how effective they were as anti-cancer drugs. It's really neat. There's a PBS special called Cancer, the Emperor of All Maladies. You can see it. It's available on their website. And anybody going into medicine should watch this. It's really enlightening. So the drugs that I worked on in the lab are still being used today as primary anti-cancer drugs. That was 16 years ago. All right? Think of all the money that has been spent on anti-cancer work since then. So what they're finding is that the new drugs might get people a couple months, a few more months of life, but it devastates them financially. What happens is their families are left, they still die. They got a couple more months with their families, but their families are left with nothing left. So this special was very good at taking a look at not only the history of cancer, but also taking a look at current treatments and the effectiveness of them. But I just find it interesting. So the drugs that I work with, they would kill you, right? You're stopping the polymerization of tubulin into microtubules. You stop cell division. But what's the difference between cancer cells and regular cells? What is the main difference?
It just keeps on
Growing faster, right? So they divide a lot faster. So the hope is that you kill the cancer before you kill the person, right? So this won't be on the exam. I'm just telling a story. I don't tell this many stories, though, throughout the stuff. But I think it makes it a little bit more interesting. But I think it's fascinating that all this money has been spent on these drugs, and there's really no breakthrough, and that these old-style cancer drugs, some that have been used for decades and decades and decades, are really still the ones that are being used mostly today. So here you can see that you have microtubules can be in the mitotic spindles, but they can also be in these circular tubes. So it's really, that's where the name came from, tubulin. So, and it is really a big part of the cytoskeleton. So I used to do electron micrographs in grad school. They wouldn't let me touch the instrument, though. I could only give them my slide, and they'd be like, back away. So you had to have like months of practice with it. All right, so this is a nice little summary of the different cell parts and the differences between prokaryotes and eukaryotes. And you can see we're not really talking too much this semester. Maybe we're looking at one pathway in plants, but you can see the difference between a eukaryote and a prokaryote cell. And again, the ribosomes give it the grainy appearance. And then you have the DNA. Viruses. I wish I had more time to go through viruses, but really they're kind of neat. They're not really living, not really non-living, so it's in between. They're genetic material. They can't live independently. So they can be either made of DNA or RNA. If they're made of DNA, they can be what we call non-enveloped or enveloped, meaning enveloped with protein. So either you have 4 different types. You can have DNA non-enveloped, DNA enveloped, RNA enveloped, RNA non-enveloped. And again, they hijacked the cells for their own use, making you feel not so good. And actually, bacteriophages are viruses, and we use them to put in our recombinant DNA into different cells so that we can use it in a good way. When I was in biotech, we had to challenge our purification process for once the cell spit out the drug, and we had to purify it from everything. We had to make sure that there was no viruses in our final product that we were injecting. So we had to make sure that there was no Chinese hamster ovary cell viruses. And so what we had to do is we had to challenge the process, add more virus than, we would do this off-site, but we'd add more virus to it and say, oh, if we can clear so much, then there's little to no chance of it being in the final product. But that's where I learned about viruses. I thought that was really, really neat. Again, we're very basic this semester. I was listening to NPR, and they were talking about how the first elements were hydrogen, helium, and lithium, and that these people with, is it manic depressive? They take lithium, and how lithium that was present at the Big Bang helps them. And they really don't know why lithium helps that. However, lithium is a lot like sodium, so they're thinking that it sort of takes the place of sodium in a certain part of the brain so that things don't travel as fast. It's a little bit more sluggish than sodium. This is what I heard yesterday on NPR. I thought it was kind of neat, that lithium that is helping so many people. But then this one woman was having kidney problems, but I thought it was neat. So other elements are formed inside of the sun and stars. In the core of the sun, we have our other elements formed. But how did life come from all those elements? Well, these elements, what they formed were proteins. And other than water, proteins are really, and having that membrane, proteins are really the basis of life. That information, okay, is in the form of proteins. So the reactions that happen in our cell don't happen on their own, all right? They need to be catalyzed. And our biological catalysts are proteins, so called enzymes. So one theory says that information coding and catalysis develop separately and then converge to form life. To support this, they found that certain types of RNA have both catalytic and information, encoding information. And really what supports this is that there's RNA-based viruses. So that does help that. This, I think, is so cool. The endosymbiotic hypothesis, and that's how eukaryotes got developed. It happened a long time ago. And what happened, and I want you to get the wording right, because the wording is really important here, that we had a large prokaryote ingest an aerobic bacterium. All right, so aerobic means using oxygen. So this prokaryote did not use oxygen. However, there was one weird one that did, so it was aerobic. It got ingested, and over time, it became the mitochondrion. And we're not going to worry about the plant cell. But there's evidence to show that the mitochondrium was from a prehistoric aerobic bacterium. And the information to support this endosymbiotic theory is that the mitochondria are similar in size to prokaryotes. They can reproduce, right? So I'm hoping that my cells do a little bit of that during my runs this semester. And they contain that DNA. Remember I talked about them containing DNA. So those are the three things I want you to cite when you're answering this test question. I'm a little bit more obvious than other professors. So that you describe the hypothesis and the evidence to support it. How are you guys with this? Any questions?
Go through that one more.
Sure, I can go through one that you're like, oh, test question. All right, so a large prokaryote, I haven't been drinking, I promise. A large prokaryote ingests a small aerobic bacteria. They live in mutualistic symbiosis. Mutualistic, so that means it's good for both of them. Mutualistic symbiosis. And over time, that aerobic bacteria becomes mitochondria. And here I have the three things that support that. It's really important that you guys have me repeat myself when you miss it, because in 2 hours and 20 minutes, your minds are going to be taking a little bit of vacation. And I understand that, and you're going to be like, oh, what did she just say? So I have no problem repeating myself. Right? Do you want me to say it one more time? Why not? A large prokaryote ingests a small aerobic bacteria. They live in mutualistic symbiosis. Over time, the aerobic bacteria becomes mitochondria. And there's the three things that tell us that this happened. The evidence to support that.